Sure. So we will have no echo.
Normally, and the microphone as well.
Okay, so let's start. Today we will be making a practical workshop on using AI, in particular large language models for aging research use cases. And what we will, so first a few words about myself. I am right now affiliated with Rostock University of Medicine, Germany, where I am actually developing applications using large language models for evaluation for different longevity interventions. Before that, I work in systems biology of aging group, where I did a lot of bioinformatics, mostly RNA-seq, but also genomics, cross-species transcriptomics, and some explainable machine learning. I am also involved in some biotech companies. And actually, most of the time that I spend now is on open source projects development. I started in this field as an activist, and I switched to bioinformatics. And right now, I am lead developer of Just DNA-seq personalized genomics platform, and of Longevity Genie, which is an assistant for biological and aging research. And a few words about the workshop. What we will be doing today. So in the beginning, I will give you some theory. So I will review how machine learning is used in aging. So most of the current major use case actually is aging clocks. Then I will look into current trends in AI, especially generative AI, and how they are coming to biology. And then I will show how LLMs and transformers are used in biology, and I will share updates on the Longevity Genie project. After it, we will actually finally go to practice. So you will try yourself how to make an AI agent, how to teach it to interact with an aging research database. And then you will learn how agents can work in a team, and also how in the future you will be able to do fine-tuning. The practical part will be made with Google Colabs. So people who are online, they will also be able to participate, and it will be done by Livia Zaharia. Yeah, here she is. So getting back to theory. So first, how can I actually hide this annoying Zoom?
Yeah, we know how to make this move.
Yeah. Yeah. So first is how AI models are applied in aging. And currently the major use case is aging clocks, and they are mostly trained on functional and physiological biomarkers. So we look at, for physiological is like our genomics, transcriptomics, our blood, different physiological measurements. But what we are also interested in is obviously quality of our life. It means whether we have diseases and whether we fit, how our health is. And In the past, by the way, I authored one of the review papers on it. And yeah, so here are functional biomarkers, some examples like speed of processing, working memory, then also some physical performance. And now we look into also important things for measuring aging clocks is that you have to know like what are the errors of these aging clocks. Typically, it's mean absolute error and coefficient of determination. And yeah, so coming back to physiological markers, so the ones that is something that everybody of you can easily try is blood biomarkers, because everybody of you probably had their blood. And so this is a picture from my review, but lately there has been interesting trends in this regard. So modern blood aging clocks, here is one of the explainable blood aging clocks papers. So you see that they trained not only to predict your age, but they try to associate it with different factors and different risks and different organs. And here you can see like like on the right top picture, picture B, you can see that like how different aspects push your biological age to the right, or blue ones to the left. And this is actually a current trend that we are seeing now. Also, we have other markers that all of you can measure. It is like with photos or with videos is facial aging clocks. There are also markers with video that you can also even check your heart rate variability. Obviously, there are some other visual biomarkers. And coming back to the most popular ones, obviously, is our omics, and methylation is the most popular, but other biomarkers appear. And we also see, so here I will be mostly focusing on trends, because in this conference, there will be talks about different specific clocks. And the trend also you see that with transcriptomic, like right down is trying to explain how different gene expressions push your biological age to like older than chronological or vice versa. And with transcriptomics clocks, when you know what genes do, it's a bit easier, but they're very inaccurate. And the most popular is methylation clocks. I actually tried it on myself. I'm happy that I'm 3 years younger than I should. The problem with this clock is that like there are huge amount of methylation points, and actually it's very hard to explain what I should do to improve my clock. And even if you look into those numerous clocks, you can see that they correlate a lot with chronological age, but not that much with each other. That means that they are connected with different aspects of aging. Often it's not super clear what is the difference. And obviously something you can try with this open source library to actually compute different clocks on your methylation data. And again, I want to focus on the main tendencies that we already have 3 generations of the clocks. So, and this tendency is, I would generalize it. It's not only in methylation clocks. I see it in other clocks as well, that we start by predicting chronological age, but it's not very useful practically, because you want to make some interventions to live longer. Then we get some clocks trained where they also looked into functional biomarkers and disease incidence. And you have 2nd generation clocks when you can actually explain which factors make you younger or older, but they are mostly trained on populations. And many friends of mine actually complained that for them, they measured clocks regularly, that it was pretty unpredictable. Sometimes like in like several clocks in different days, like have like 1 or 2 years difference on the same person. So you don't know if like one one day can make you appear younger or older, something is wrong. So they are good on population level, but they're not on individual. And third generation clocks, they try to look longitudinal data sets, so you can train it to be more or less robust for individual person with multiple timestamps. And in other clocks like blood clocks and so on, people try to actually follow the same trajectory. And going back to existing trends, let's look into actually using of different machine learning models. So here is actually a history of evolution of machine learning. And the first machine learning, actually, probably majority of you did some machine learning called linear regression, when you tried to fit some data to a line on statistics. And with time, we got new and new concepts. The interesting part of it, if you look into many popular methylation clocks, for example, that, for example, Horvath's clock is elastic nets that technically linear regression on steroids. So linear regression and a small trick with regularization. And many other clocks, for example, for blood aging clocks, the most popular ones, they use gradient boosted together with decision trees. So things like LIGBM, XGBoost. So it's actually algorithms discovered originally in 90s. And if we look into more modern stuff, that we see that for visual things like facial aging clocks, video clocks, where you have recognition, people are using convolutional neural networks, and recently they are switching to vision transformers. And there are also some experimental aging clocks, actually some of them by in silico medicine, who will also present in this conference, that are trying top-notch models, and they also trying generative models to predict how things can change if, for example, some genes would express differently or some drugs would be taken. So the question is, why are the majority of the clocks are still lagging behind in terms of models? And actually, when you look into the structure of available data, it's not really surprising. So when I came to bioinformatics, I had this assumption that everybody was talking about this big data. I thought, okay, biology has a lot of data, so it will be possible to use the best deep learning models. In reality, it's actually, to a large extent, a myth, because the data is big in terms of file sizes and in terms of dimensionality. We have like 40 or 60,000 of genes, depending on sequencing type. We have also a huge amount of proteins, 20 million methylation sites. But the issue is that the number of samples is very small and way smaller than the number of features. That means that a typical rule of thumb that the more powerful model you have, the more number of samples should be larger than number of features. Otherwise, it would just overfit and will always find some way to make non-meaningful predictions. Yeah, so this is the major problems. And unfortunately, when you have hundreds of data sets, hundreds of samples, you cannot train something really fancy, but things are actually changing. So in terms of the recent trends, there was this RDD conference papers on actually summarizing things. So stuff that I was talking, it's also reflected is actually different. So we see use of some generative model, and one of the reasons, and also a huge use of explanatory AI. And So a few words about explanatory AI is actually this in this slide you see typical attitude to a person who got his methylation clock, for example. So you see some number and you actually don't know what it means. Is it like I am not working out enough, I'm older, or I am my diet is wrong? So you see some kind of black box prediction. And to mitigate it, and what you actually saw in some of the recent aging logs, that they trained so-called explanatory XI models that just take your major model and play with it in order to understand which features are important. So here in this picture, you see actually comparison like black box models that just somehow detects cat and you don't really understand how. And another model that it's explanatory, it actually tries to define some features, if it has whiskers, if it has tail, if it has claws. And this is way more useful when you want to make a decision. And by the way, the whole explanatory AI movement was pushed hard by DARPA. That's kind of interesting. And one example that is very illustrative of this is that the way how machine learning model makes recognition is very different from our human intuition. And here it's a one pixel attack when people analyze the model and discovered the most used pixels. And they change one pixel and it turns horse into frog, deer into airplane. And for humans, it's almost impossible to see what changed because humans look into some reasonable features. And we try to, because we want the model to have actionable, meaningful features, we actually try to understand how models makes decision and connect them with something that makes meaning and that we can change. And here are some examples for explanatory models in medical diagnostics. You see with color, it shows which region is the most important for the model. Here is from one of the blood aging logs that you see you have like a base age, like it's, this is a method called Shapley additive explanations. It's like it assumes that you have a base age of say 48, and then it just some features like total cholesterol is probably bad. It says you're 3.5 years older, and some features, for example, like your glucose values, they are better than are needed for your age, so they decrease your prediction of biological age. So this is a local explanation of specific decisions. There are also globals that explain general behavior. And majority of aging clock now that are recent actually using some similar techniques. So yes, so I talked about explanatory AI, but obviously the most interesting trend now is actually large language models and all those fancy generative diffusion and other models. And what actually changed, why we finally starting to get some data. So you see that biology and medicine, it had like a long way, and it was pretty hard to train something like with several hundred samples to make some deep meaningful predictions with like 40,000 features. But because of single cell data, we are actually getting way more data because even though it's expensive, you can spend like 4,000 euros on one sample, but you have millions of cells and you know knowledge on a level, on a cell level. That means synthetically you can think about each cell as a microsample. And that means that with this, if you have 2 million cells, and for example, you have on this, because you detect less genes, for example, you had like 10,000 genes that you stably could detect, it means 10,000 features and 2 million samples. And suddenly you actually can train fancy deep learning models, and you are not bounded by this limitation. And what actually it leads to that in the past, we had this trend of hypothesis-driven search when we had some idea that something influences something. We gathered data, we connected it, we made some study, we checked our hypothesis. It's pretty robust, but it's like a lot of effort and a lot of time. And now when we are actually getting large models that can make their predictions, we often go like in more agnostic way. What will the data tell us? So we see what interesting things we get out of the model, and then we screen them, and then we see which of them makes sense, and then we investigate them already like in a hypothesis slow but more robust way. And same second part is that we are switching from correlations that people wrote a lot about, like connection of your coffee and lifespan. Or for example, there was this interesting thing with methylation clocks when people checked correlations in terms of improvement of biological age. And kidney transplants or RC therapy were the best in many methylation clocks, simply because you do kidney therapy when you're already bad, and like a kidney transplantation. And obviously, for people who need kidney transplantation, their methylation age predictions will improve a lot. But it doesn't mean that all of us have to transplant kidneys to become younger. And yeah, and also many classical methods with multiple statistical analysis, they change into using different generative models and assessing their behavior. And the way how we are actually dealing with this large amount of data that we get from various sources, obviously, the major source is text that we see in large language models that are trained like on trillions tokens, but also with single cell RNA sequencing and some other modalities, we're also getting larger data. The way how it's trained is actually in several steps, because even if we get like 20 million cells, we don't have like labels what the cell do. So we first have to learn, it's called pre-training, a compile or training foundational model, people also call it this way, a compact representation of the domain. And well, for example, if we take, I will just imagine we have like a rectangle, like, well, here is a phone. Imagine we are just training something on geometry. So if we look into pixel by pixel, well, I don't know if you have like 30 million pixels phone, it will spend like a lot of memory and a lot of 30 million parameters to actually start understanding how it behaves. But if we, for example, dealing on a geometrical way, and we learned that it's actually some parallelogram to describe its shape, we just need what is the width, what is the height, what is, if it's 3D, what is the depth, and maybe some other things like color. And it means that when we do this pre-training, trying to understand how data is structured, we get way smaller number of parameters. And on top of those parameters, we can already, because we have less parameters, we can train in a classical supervised or semi-supervised way, and we can get better understanding. And we can also even generate some synthetic data. And the way, and also, so here I had like these examples with in biology when we think that every cell is a sample, and we try to somehow predict the expressions. But if we talk about what is closer to us, the words, if we take any large language models, is actually, well, formally it's a decoder based transformer model. That means that you get some input, and every steps, it just generates a new token. Well, it's almost a new word. And when it actually pre-trains, it gets text. When you just delete some, it's called masked training, delete some words, and you try to predict those words out of context. That means that you don't really need to label everything. You can just learn the with attention mechanism, the representation. And here, for example, on a picture, you see a representation of different words. It's called embeddings. So it's technically they have multi-dimensional space where if model is trained, it recognizes that cats are closer to dogs than bananas. And you can even do some arithmetics, like, for example, like queen and king and men and women. And you can see that like the relationship, if you add something from women to a king, it will be queen, like semantically speaking. And it's also used in a technique called semantic similarity search, when instead of searching by keywords, you search by similarity in representation space. And often it can understand totally different synonyms that have used different letters just by how close they are. And here is how we do it with, so here are two ways how things actually, it happens in all LLMs, that you use either autoregressive or masked modeling. That means that you just use part of the sequence. So you split the sequence into tokens, can be nucleotides, can be like specific genes, and then you substitute some, you try to learn and substitute something that you delete by surrounding. And important terminology here is when you train this compact representation, is then you make a second step that you can train it to do something useful. Because when it learns representation, it will be way easier to have for even like supervised training. Like, for example, what will change if I will knock out the gene? So if you have a good pre-training, you will need way less data and you will get way better accuracy if you will train on tasks of your interest. And here is example from GeneFormer. It's very popular single-cell RNA-seq foundational model. Then it's when zero-shot learning, like when you just, it's pre-trained, it already can be a bit useful when you try to describe processes like some reprogramming or differentiation. But if you want to do something useful, for example, you're doing partial reprogramming and you want to understand which dosages of Yamanaka or some alternative factors you have to give to actually push the cell state, you actually need to take foundational model and give it additional supervised or semi-supervised data, like what happens when I add, what will be there in a seq when I add this transcriptional factor. And with this way, it actually trains. So on the right, you see like how GeneFormer, what useful stuff it can do when you train it after fine-tuning. With large language models, it's also, if you look any foundational model for human use, it's pretty useless. Because what it was learned, it's like takes part of Wikipedia, you delete some words, it predicts missing words. So if you give like half a page of Wikipedia, it will predict you the last half of Wikipedia. That means that it's not what we really want. We want like smart chatbots. For this reason, people use fine-tuning, including methods with reinforcement learning that actually turn it into like producing some useful dialogue-based output. So here are some examples from GeneFormer. And also important part that is actually similar both for biology. So here I am just trying to show the similarities. So in both cases, when you pre-train some model and then you fine-tune it for your task, for large language models, fine-tuning can be like answering chat and giving something useful. If you want to improve its performance, you can either improve your querying, what you ask, or you can do additional additional post-training by like tuning it with either supervised data or with some feedback, like you like this answer or not. And typically, the best thing to try is first to do. You shouldn't start fine-tuning until you will understand that I did everything I could with prompting. That's like a general rule of thumb. Because prompting is often way easier, and you actually understand what are the problems that you have to fine-tune. And there are some methods that actually allow fine-tuning. You don't need to be OpenAI to fine-tune it, because there are some parameter efficient models like LoRa methods, LoRa or some adapter base, that you don't train the whole multi-billion parameter models. You train a subset of it for your application. So if we will have time, actually, Livia would show how fine-tuning works in practice on your actually personal computer with a small 7 billion model. And also, overall, if we talk about model, we also have the context that you have, like, it's integrated into some application where it is useful. And you, when you develop it, you think what you develop it for. If you need to develop, or you can, majority of models are actually just fine-tuned on top of popular open source models like QN or Llama. And then people just adapt or merge and adapt to their task. And in many cases, it's actually even like ChatGPT, there are ways to fine-tune it. So in many cases, developing a model from scratch is actually big overhead that you shouldn't do. And the last part is actually how you will use it in your app, because models can give useful output, but they can also orchestrate, and with help of models, you can have autonomous agents that call functions and do some job. And the last, in terms of connections between LLMs and biology, is actually really interesting paper on cell to sentence, when people said, okay, why should we invent the ways to specifically describe biology differently if we already have good models that work with text? Maybe we can just get, like here on the picture, you see that it just spit out the text of the genes, and then it say, okay, I will take general text model, and we'll just train it to spit out RNA expression phrases. And surprisingly, this approach actually works. And yeah, so here are like more details how they do it, that they just train it as if it's a text model. They just, instead of regular text, they just textify the expression matrices, but they train it as if it has like masked parts of the text. And so this is like how they do it, a sentence to genes. And in, for example, gene former, about which I talked, they did a bit differently. They had a specific way that they were afraid, because biology data is extremely noisy. So they didn't look into exact expression values. They just looked into ranking, like which gene is more expressed than others. So they actually just had a sorted list by expression in rank. So it's like first, second, third, and then they just tried to predict the rank of a gene that is masked from the surrounding. And the challenges overall of training new foundational biological models is like you have to, so to fine tune, you don't need that much, but to make foundational model to pre-train it, you need petaflops of computation, and not many labs can afford it. And the other problem is that it's still like very early stage, and still the amount of data is growing because of single cell sequencing and other trends, but it's still very small, that often you actually don't have enough data. And actually there is a way, when you read any papers about another LLM generative foundational model, you can understand if it makes sense or not. If somebody tried to use foundational model, you needed to represent biological processes. If you made this representation and you trained on top of this representation, and it is better than you trained without any representation, then it makes sense. And as you see in this chart, in actually many cases, majority of them are just additional complexity, and they don't really improve results in terms of if you just train the data directly, like some prediction of something on the raw data without these models. It can be similar in accuracy, and obviously it will improve when there will be more data. And in terms of like dealing with models, so Livia will show it, but to let you know that most of the non-proprietary models are on Hugging Face, and there are different leaderboards. In Rostock, we are actually considering building some leaderboard about answering aging research questions. So if anybody of you who has medical background is interesting, we actually would have some querying. So far we started with like medical things, whether like a person should, for example, take rapamycin at some dosage, because here we can check for toxicity efforts, but we will consider doing also other relative measurements. And one thing to mention also, there is this popular trend where people try to, those so-called biohackers, they try to like measure everything about themselves. And there are many startups that try to make so-called digital twins. It means that if you told the model everything about you and it knows everything, like what's your weight, what did you do, it can actually answer your personalized questions better because it has it in the context. And the last chapter is actually our developments. Actually, one of the main things that I do is this longevity Genie open source project that, by the way, is supported by HEAL. So thank you very much, Didier. We actually started it for pragmatic reasons that like a year and a half ago when ChatGPT-4 became a thing, and there was like boom of models, and it still actually continues. We wanted to get better answers on aging-related questions. And in my case, I started just trying how it will answer genetic questions because we have a second open source project, just DNA-seq, that can generate your reports and make some analysis in an open source way for longevity genetics. And by that time, actually still now for many genes, it actually gives really bad results. So for ChatGPT and other models, they are usually trained on the most popular data. So it's a problem of short neck, long tail, is that it is really answering well on popular topics like, for example, APOE, it knows more or less well, even though actually it makes mistakes sometimes. But if it is like cutting edge research that is not talked a lot and not properly indexed or is meaningful for a small number of people, they know it pretty bad and they make also a lot of mistakes. And yeah, so here are examples of some of our like internal tests with this. And also there are issues when, so LLMs are also like humans, they are bad in counting numbers because actually of probabilistic way how they are trained. So they actually have to generate Python code to actually do serious mathematical computation. So at Hackathon, like a year and a half ago, we actually developed a chatbot. By that time, we did it on top of ChatGPT API. Now we actually support multiple models, including Llama, including ChatGPT, including Claude. And we taught them to generate queries to databases, actually something that will also Livia show you how to do today. And it had like bad results in the beginning. And then with some improvements, we actually got really on point results. And okay, let me skip to some technical details. And yeah, and we had also custom GPTs that people could use with just regular custom chat GPTs. And now we have this longevity. I can actually show you. So we have longevity genie website. So if you ask like about specific, for example, allele, what it does, it actually makes a query to our genetic databases, and it gives way more information than regular ChatGPT. And we have like multiple. So far, it's very hard to make everything as one model. So we have like multiple models. Technically, it's some agents that are run by ChatGPT Mini or other models. And so we have genetics genie. We also have one on OpenGenes. Let me show. So OpenGenes, it's actually what Livia was working about. So if you can ask about like what, oh, I think, okay, I will check the model. I think something was wrong with the update. Okay, let's try here. Yeah, so if you ask, for example, yeah, because we have like two deployments. So if you ask, for example, what extended lifespan in mice of expressing VEGF, you get some values. But if you ask the same like regular chat GPT, actually, I'm curious to know. So it made some search on a gene, but you see it doesn't have any numbers. That means that it doesn't, one of the most important things that you want to get is like what extends my lifespan. And here actually it, so there is this OpenGenes database, and it provides information about longevity associated genes and how much they extend lifespan. And in our case, it can query this database. So we have a lot of questions about like different genes and different interventions, and even some gene variants that regular ChatGPT without databases barely knows. And the other thing, let me actually show. The other thing is that you can, so here we are using large language models to call different functions and to do different actions. And it can actually interact with specific models trained on biological data. So here is actually, okay. So there is, let me first explain, and then I will show some info. So here are like I showed for ChatGPT for different genetics and longevity related questions. We also have integrations to obviously just DNA-seq platforms. So in genomic reports, you have LLM summarizations. And yeah, and we also have integrations. Let me go to specific. Okay, let me see. So we also have integrations with some generative models like Precious GPT. They have by in silico medicine. So they have a little bit like in their deployment, it takes them some time to okay, so looks like their endpoint is temporarily non-responding, but yeah, but it's on their side. And I will later restart it and show how actually it works. And another thing, actually, So we also have learned a lot. So we implemented, so I showed you some databases. We actually, in bioinformatics, we have a lot of databases, and obviously you don't have, if you have a small team, you don't have much of researchers, but much of resources to implement everything. So what we have done is that we developed a bunch of modules, open source libraries, and the way to extend. That means that, well, Livia will show some stuff, how things work, but it's very easy if you have a database of your interest, that is like biology, aging, or totally unrelated. It's pretty easy to add an agent that can also query this database, and it will use ChatGPT, Llama, whatever model you want, and will also get some extra data from the database that is not indexed, because databases are typically not indexed by large language models. And we learned also several things that if you put too many databases at once to like one model, it starts like confusing. It's like a human with too many choices. And now we are developing a two-stage approach when we have first a router model that decides which databases are needed and then selects, talks with the agents that know those databases. It's not yet deployed. However, you're also using another technique called knowledge graphs. And the idea of knowledge graphs is that you just merge many databases together in like a knowledge graph, and you query as if it is one database. So we have, we are a member of BioCharter Consortia that is actually working on this technology. So on the picture, you see that you have different adapters that convert different resources and feed in one large Neo4j database. So your LLM will just query one database. And here are example of drug interactions of metformin. We can also call other models. So I will restart to see if Precious GPT is up again. But here is an example of Gluformer. It's actually a model trained by Livia on top of glucose values predictions. That is useful not only for diabetic, but it might be useful when there will be enough data for healthy people to track their diet and sports. And yeah, and let me see, let me check if Precious GPT is back. Okay, I hope it's not cached. Okay, it looks it's actually, so they, let's run it. So it looks like it's running because it usually takes time to get an answer from them. Okay, as soon as it will finish, I will give an example. So, oh, wonderful. So I will show you now. Yeah, so they had this configuration. If you don't query them for a long time, they just freeze it. And usually you have to wait until it gets active again. But here it's actually worked. I asked, could you please provide me with the expression profile for a 70-year man? And it actually generated a synthetic which genes are expected to be upregulated and downregulated for such parameters. And we integrated it with, so let me show another query. They integrated it with standard bioinformatic tools like gene set enrichments that can actually explain what those genes means, because the model tells, okay, these genes will be activated and this will be downregulated, but usually you look at higher level like biological processes. And here I, for example, ask another query. It's more interesting that if it's if it is how skin proteomics data would behave if a 30-year-old women are taking metformin. And obviously for non-like experts, just names of the proteins are not useful. But after enrichment, you can actually see that you have some estrogen signaling that is upregulated, probably good. And then you have some downregulated UV response and apoptosis, that's probably bad. So here is like a screenshot that explains it, shows the part that we got from Precious GPT and our like enrichment tools. So we can ideally, if we have enough tools, we can connect multiple databases and multiple models together. That means that if you have a project, it like checks, okay, I have to call this library for analysis, and then I have to use Precious GPT to predict what will change. And then I will see which processes are involved. And then you can also get some data from database and some text to have like some ideas and decisions that can be interesting to investigate and validate if it's true or not. And yeah, and actually regarding Precious GPT here, they actually have, I think that there will be in Silico talk today. So I just briefly mentioned that they trained pretty small actually transformer model on expression data and knowledge graphs. But it's actually good at small because it's easy to run. You don't need supercomputers, and it gives predictions that if you plan how you want to extend lifespan or something, you can just say, what will do if I give this drug and overexpress this gene? And it's the way how, actually, I totally agree with their vision, is that you can make a lot of simulations to get like interesting idea from the data. It's like data-driven hypothesis. Then you can do some screening. And in Silico, they have robotics lab for it, but there are other things like for C. elegans, there are those automation machines, for example. And then you use some LLMs, and in Silico, they have Dora, for example, that are integrated with papers to actually summarize things together. And yeah, and coming back, yeah, so that's what we are working on. And overall, now we are actually working on also multi-agent systems, like you can split everything like a team, that you can have different engines like assimilated researchers, that one does criticism, another does research, another can call models or write code. And actually this approach worked well in programming, and we are also making things extendable. So if you want to have an LLM chatbot that can run your tools or analyze your data or answer about your databases, we are open for collaboration. And I think with this, I already overloaded you with new information. Yeah, just want to briefly mention like the team behind the project. And I think the best way to understand things is actually to try some simple things yourself. Yeah, so here is the team involved. And with this, I will give a word to Livia to actually show some basic things about LLM agents.
Maybe first, Anton, if there are questions